Okay, so the time just struck 5 p.m. Central European Time, which is the official starting time of the webinar. So I would like to say a very good morning, good afternoon, good evening to you, wherever you are in the world. Thank you very much for joining this webinar. It is my pleasure and privilege to actually open the first webinar from NOUL. It's great to see you all here, and thank you very much for taking the time to join us. My name is Jack Taylor, and I am the Global Product Manager for Malaria at NOUL. And today I will be delighted to be acting as your host for the Malaria webinar entitled AI and Automation in Malaria Diagnostics, and how we can enhance accuracy and efficiency for both laboratories and field sites across the world. So before we jump into the slides, I would like to go through some administrative tasks. There is at the bottom of your screen a Q&A feature or functionality. And so I would like to request that in case you have any questions that pop up during the course of the webinar, please feel free to enter the questions into the Q&A section below. You should see it in the bar at the bottom of your screen when you move your mouse. And then we will answer all of the questions to the best of our ability at the end of the webinar. In case there are any questions that we cannot answer, we will, of course, follow up with you directly to the webinar. On top of that, there will be at the end of the webinar a survey for you all to fill out, and I would like to encourage all of you to do so, please, because it really helps us to improve our webinars for the future. Your feedback is very much valuable, and if you fill out the survey, you will receive also the PowerPoint presentation together with the recording from us. So without further ado, I will jump straight into the table of contents today, which you see now on the slide. So first of all, I will give you a short introduction to NOUL. Who are we? Where do we come from? What do we do? What is our mission? And I will then introduce our speaker for today, Professor Hans-Peter Beck. And then I will hand over to Professor Beck for the remainder of the presentation, where he will give an overview of malaria diagnostics in Europe, MyLab NOUL system, which we focus on today, why you might choose the MyLab NOUL for your laboratory or field clinic. He will also present some clinical data and the overall value proposition of our system. And last but not least, as I mentioned earlier, we will have a question and answer session at the end where we will do our best to answer all of your questions. So, about NOUL. So who are we? So NOUL actually means sunset in South Korean, the Korean language, and that's the reason why you will see a lot of orange around, and because orange is, of course, the color of sunset, in case you were wondering where the name comes from. So what is our mission? Why do we do what we do? We explore global challenges that threaten human health and life, discover novel potential solutions, and realize those possibilities together with our expertise and technology. Our official company name is NOUL Company Limited, and we were founded in December 2015. That makes us almost 10 years old to this day. We were listed on the Korean Stock Exchange, specifically on the KOSDAQ, on March the 3rd, 2022. Our CEO's name is Chanyang Lim, and we currently have a total investment fund of $85 million US. We have more than 150 people working for the company, both in South Korea and across the globe. And we are located in our headquarters in Yongin in South Korea. That's just south of Seoul, for those of you who don't know. Our headquarters is made up of two main divisions. It's the headquarter office, so the main marketing and product strategy staff. And we have also the research laboratory where a lot of our R&D activities occur. And we have the manufacturing plant where we manufacture both the instruments and our cartridge system, which you will learn a little bit about more in detail later today. So what exactly do we offer from a product perspective? We offer the MyLab device, and on the MyLab device, we have three possible applications. The first of which is a peripheral blood smear analysis application. The second is a application for screening of cervical cancer. And last but not least, and the application that we focus on today, we have a malaria diagnostic application. Professor Beck, I think the screen is sharing the presenter mode.
Oh, that should not be the case. Okay, sorry. I'll just change that quickly. So that should be better
Mhm.
now.
Perfect. Thank you very much.
Sorry.
And the next slide, please. So this slide gives an overview of where we came from, where we are, and where we're heading. So our road to global success. In 2018, we gained our ISO 13485 certificate. This is a quality management certificate specific to in vitro diagnostic devices, as you may well all know. We also received our Good Manufacturing Practice certificate in 2020. All of our devices and applications are CE IVD marked, which means that they can be marketed and sold in the European market and the European Economic Area, as well as a few additional countries that also follow these regulatory landscape. We also received recently the Medical Device Single Audit Program certificate, the MDSAP, and we do have many other global certifications in progress. A list of countries you can see here as an example of some of those where we are currently in the registration process. With regard to global recognition, you might well recognize some of these organizations that have looked upon our company favorably, and we even won some awards. So the first of which was the People's Choice Award in 2019 from the Gates Foundation. We were awarded a grant in 2020 from the Wright Foundation. And 1 year later, we also announced an on-device partnership in 2021 with an AI company called NVIDIA. I'm sure you're all well aware of them. We introduced our malaria diagnostic application in 2022, followed 2 years later by our cervical cancer screening application in 2024, to much acclaim by an organization called Unitaid, which Professor Beck will explain a little bit more about later in the slides. We have many strategic partnerships with leading laboratories, pharma innovators, IVD manufacturers, and more organizations around the globe. And we've highlighted just four of them here. Some of them you may know. The Limbach Group is a large private lab and clinic chain in Germany. Neon Coden is, of course, a well-established IVD manufacturer. Seegene Brazil. Seegene is again a South Korean company that is also an IVD manufacturer, specifically focused on molecular biology. And we have installations in, for example, the Charité Hospital in Berlin, amongst many other partners around the globe, and many more to come. So we have partners with over 100 organizations across 25 countries, while conducting at the same time research and operations in more than 20 countries around the globe. As I mentioned earlier, our headquarters is in South Korea, in Yongin, just south of Seoul, but we also have a regional office in Switzerland, and NOUL has local staff in Germany, the UK, France, Mexico, and Brazil, and many more to come, we hope. But without further ado, I would like to introduce you all to our speaker today. Our speaker today is Professor Hans-Peter Beck. He is a Professor Emeritus of Molecular Parasitology at the University of Basel, and he also led the research unit at the Swiss Tropical and Public Health Institute for 25 years. He has expertise in the molecular processes of tropical diseases, specifically focusing on malaria research. He was a key contributor to establishing molecular genotyping methods in clinical and control trials for anti-malarial drug development. And he has also co-authored a very impressive more than 150 peer-reviewed scientific articles, which actually gives him a H index of 54, which I'm sure you'll all agree is very impressive. And without further ado, I would like to hand over to Professor Hans-Peter Beck to give you an introduction to how his story started together with NOUL. Thank you very much.
Well, thank you very much, Jack, for the introduction and for giving me the word here. Good morning, good afternoon, good evening to everybody who's listening from around the globe, hopefully. It's a pleasure for me to be here to speak on this webinar for NOUL. I just would like to tell you a little bit about the history I have with NOUL. I came in 2015. I came to Korea, to Seoul, in a government mission from Switzerland to see what is on what's the startup situation in South Korea. And during this visit, I visited the headquarters in those days, which was a small office somewhere in Korea of NOUL, which has just been founded more or less. And I was already very much impressed by the vision and by the idea, which I thought is exactly what we need in our work in malaria research. And I returned in 2018 again, and I could see there was a significant growth in the company, and they had now a very impressive proof of principle device. And it convinced me that this technology has a future for malaria control and malaria combating malaria. And today, I'm scientific advisor to the company, and I'm still convinced that the device will be extremely useful in the future in our endeavors to curb malaria. And I will tell you a little bit about the whole thing. And before I do that, I will tell you a little bit about malaria research in Europe, what's happening here. Um, the European Union and the European Economic Area region was declared malaria-free by 2015 when the country Georgia finally got its last transmitted malaria case. However, malaria is still a problem in Europe because we do have a lot of migrants and we do have a lot of travelers, and we do have now, through global warming, we have the vectors which could transmit in principle malaria. So there could be a danger that malaria is coming out again within the European community. But on the other hand, as I said, we have a lot of migrants and we have a lot of travelers, and we need to have a proper malaria diagnosis. On the other hand, malaria researchers are still quite important, I think, in advancing, treatments and vaccines and diagnostic technologies for malaria. And we are supporting very much, artificial intelligence diagnostics, in principle, to support the malaria elimination program of WHO and other players. And we know from that one, we know the diagnostic platforms remain a crucial piece for research and surveillance, even in places like ours here, where we are malaria-free. So we want to contribute to WHO's malaria elimination goals in 2030. We want to play a central role in developing new technologies, therapeutics, vaccines, and diagnostic technologies. And we are pioneering next generation malaria diagnostic research based on IE and molecular technologies. That's why Switzerland is hosting an office of NOUL. And with that, we want to strengthen the global surveillance systems, investigating resistance mechanisms, and we want to prepare for vector expansion because of climate change, as we all know. And the problems we face here in Europe is because we have so few cases, comparatively few cases, we lack of highly skilled specialized personnel. So we have only few clinicians with hands-on malaria diagnosis experience. So let's say a patient comes, we don't have personnel who can read the slides properly, and they lose their information because they don't see it very often. We have very few samples, of course, which leads to this lack of skilled personnel. And finally, we need to have the diagnostic infrastructure because we are getting these imported malaria cases, and we have to diagnose them quickly and accurately to prevent any kind of delay. So here I have a chart from 2013 to 2022 of malaria cases happening in the European Union and in the European Economic Area between 2013 and 22. And we have about between 8 and 6,000 cases a year. We see the dip here. This is the corona epidemic where nobody could travel. And we have all confirmed cases. Most of them were imported malaria cases through migrants and travelers, but we had already 104 locally acquired malaria infections in this time period. The mode of transmission is local transmission, health acquired fish infection by blood transfusion, for example, airport malaria, mosquitoes being transported, and other modes like lab acquired or something like that. And then in 19 cases, it was not determined. So I'm not saying it's a great danger, but we do have a danger of malaria being resettling in Europe as it used to was, as it used to be in the 50s, even we had malaria here. But anyway, so what are the tools we have at hand for malaria diagnostics? We do have the classical, still gold standard microscopy. I don't have to explain microscopy to any of you, hopefully. You know exactly what's the problem with it, you know exactly the quality of it, and you know what are the weak points. One weak point is in particular in every area, often the required skilled personnel. I'm sure you all have experience that you have microscopists who are not at scratch as they should be. So the required personnel, the low sensitivity, and in principle, low portability is an issue with it. Of course, you can see the stages, you can quantify, and so on and so on. Then the next level is, of course, the molecular tests, PCR, LAMP, which is highly sensitive, can specify, can quantify, it's all fine. However, it's expensive. It requires a high cost infrastructure. It has a complex workflow, and it is not doable in the field right now. Then, of course, we do have the RDTs, very much promising RDTs. It's okay. They are very field applicable. They are rapid. They are reasonably cheap. They are in principle operator independent. But they do have one big problem, and this problem is increasing. That is the false negatives and the low sensitivity. And the false negatives come from the HRP2 deletions, HRP3 deletions, which occur. And most of the RDTs are based on HRP2 detection. So if it's deleted, if the gene is deleted, there's no product and it cannot be used for identification of the parasites. And finally, we have a very small possibility, which is a fluorescence detection. We have one instrument actually, which is available commercially, which provides rapid results. It's a Sysmex system, but it has high costs, needs good infrastructure. It does not discriminate between the species or gametocytes, and it doesn't show you any kind of morphology. So and so far, I would say, it's a niche product, and it's not really applicable for the field. So, but where are we now with NOUL, with the digital microscopy? The conventional tool we know is microscopy, PCR, RDTs. Our tool, the NOUL MeLab, is a digital microscope. It is artificial intelligence-powered automation. It reduces dependency on an operator. It has a reasonable high sensitivity. It gives us a species, and it identifies the stages. It's portable. It can be used on site. You just need a power socket. It can really be used in the field, and I've seen it being used in the field, and it could well become the gold standard on the WHO Global Technical Strategy for Malaria 1916 to 13 quotes it as a supporting element of the strategic framework, harnessing innovation and expanding research. So it's very clear the MeLab product falls into that supporting element one of the strategical plan. It also has been mentioned in this in the strategy, in the WHO Global Malaria Program operational strategy, it's on 2040 to 2030, as a tool for diagnostics. It's a preferred product characteristics for digital microscopy in malaria. So it's very clearly, it's very well aligned with international demands on diagnostics in malaria. It's very clear. So the Me Lab, how does it work? What is it? Perhaps some of you have seen it already. Some use it probably already, but some may not have seen it. It's an all-in-one platform which transforms malaria diagnostics. It's a digital microscopy. It's a digital microscope which replicates the classical diagnostic performance of WHO certified level 1 microscopists, but it maximizes efficiency through a workflow automation and the artificial intelligence powered analysis. What does it mean? So we do have the machine by integrating this solid-based staining technology, which is proprietary on the device of AI. So we make the smears, we make already a malaria smear and staining in the machine. Then the whole process is automated from smear preparation, as I said, and results in the and ends up with the results all on a single platform. You have to do nothing else than put a single drop of blood into the machine. So it provides an automatic entire workflow. It is powered by an on-device artificial intelligence system. The system detects abnormal cells and identifies all the malaria species. It can view on a remote monitor for via a network connection. So this is in particular important if you're unsure what it is. You may send it to somewhere or somebody else may be looking at it, which is a fantastic option you have. And secondly, or lastly, we have a qualitative and quantitative analysis, which enables a diagnosis with only 5 microliters of blood, as we usually have it with a classical microscopy. Below here, you see what is really needed. So you have this instrument here, which is a handsome little instrument. It's portable, you can carry it, it's easy. You have the cartridges, which you need, and you can link it to this viewer where you then can see in high resolution whatever you have seen in your malaria slide. So the core technology of the whole thing is the next generation staining and immunostaining. It is proprietary to NOUL, and it is very importantly, it is a liquid-free staining technology which was developed in-house and is patented. What does it mean, liquid-free? So you don't have any fluids which are flying around with all the dyes and whatsoever. You all know that in the classical way, they have methanol fixation, drying, staining, washing, drying. So it's all a fairly tedious process. And it can go many things can go wrong. As we all know, if you have Giemsa stain is getting off and on. It's not good quality sometimes. You get precipitates in the Giemsa stain. You all have this knot here. So you have no liquid in except the fixation, which is easy. It's an ethanol fixation. But everything else is in a dry system. You have a single cartridge, and the whole staining process is performed very efficiently and very reliably. And on this platform, it significantly enhances the performance and is very convenient for the user because, as I said, 1 drop of blood and the machine does it all. So here you see, this is a manual process. It's tedious, it's long-lasting. If you go to the NOUL machine, you have this NGSI, and you see it blood in, slide in, and you've got all the data and after 15 minutes or so. And you get that because the machine, after having taken all these digital photographs, it's got the proprietary artificial intelligence in the machine itself, on the machine. So you don't need actually an internet access. It's all on the machine. It's optimized for the Me Lab device, and it automatically detects the target cells within a blood sample. And it will then give you a final infection status. It will give you the species of the infecting malaria parasite, and it will give you the level of infection. So it is positive. Negative, it will tell you about the gametocyte detection, whether there are gametocytes or not. It will give you the malaria species, and it will give you the parasitemia. So it will give you a percentage of infected red blood cells. And it's all happening in this little machine. So you adhere, load the blood sample, insert the cartridge, and then everything happens. It smears, it fixes, it stains, it does the imaging, it does the detection, it does a classification, and on the screen later on, you will see the patient report. So you know exactly what's happening inside. And just to let you know, so at a global conference, NVIDIA has selected, the NOUL device as one of the 5 notable technologies at this conference in 2021. So it has been already recognized. So one again, it's an intelligent automated workflow. It does everything, the entire microscopy, automatically, slide preparation, staining, sample analysis, result generation, and everything can be handled on this device, streamlining the workflow and enhancing the efficiency. So what we do is here you see the cartridge loaded with the blood, smearing, fixing, staining, and then we have the digital microscopy. Here we have to say the digital microscope is not doing just one image. It makes about 10 stacks per individual image. So you have the option on the machine later to scan and screen through the height of the microscope as you would do it in a normal microscope. You would have the different levels of focusing with these 10 stacks you generate. Then you have the on-device AI analyzing all the blood samples. Normally, it's about 200,000 blood cells which have been analyzed, but that can be changed. And after 15 minutes, you get the results screen here on the machine itself and on the viewer if you want to. This here is what you get if you have it in the laboratory standing. You have all these little pictures here, little images, which tell you exactly what you're seeing. So this is now the field you're seeing. A cell which the machine has recognized as special or as infected. It's been highlighted, and on a small image here on this side, you can actually look at this individual cell and confirm whether it's a true parasite as you expected. The same is here with gametocytes, very clearly highlighted gametocytes. You can see that, and you can see that. So it always gives you a consistent result and it proves the research capacity. It can be achieved. It's very important for clinical trials. I collaborated a lot with pharmaceutical industries with their clinical trials, and they really needed such a device, and it's been used now in clinical trials. And this is because it's so reliable and it's all point of care. And in each of the reports, the cells are classified as species and stages or as normal RBCs. If you see something which you think, oh, that's probably not a parasite or so, you can reclassify it on the viewer. So you can say, well, this is probably a mistake, and then you can improve the result even further. So one of the performance matrices. So MENAP demonstrates a high performance detecting even low-level infections with minimal misses. It provides an accurate and reliable diagnostics that support timely treatment and contribute significantly to malaria controls. So we have an LOD of 43 parasites per microliter, and that is based on scanning 200,000 red blood cells. As I said, this can be changed. You could also, change it to 400 red blood cells, but then the time takes much, much longer to be analyzed and to be scanned. The sensitivity overall is 98%, and the specificity so far is 100%. And internal data indicate that the limit of detection could actually be 43 parasites, as said. But on clinical evaluation, the MELAB demonstrated the capability to detect parasites at concentrations down to 28.5 parasites per microliters, and the clinical cutoff was then determined as 25 parasites per microliter, always based on these 200,000 RBCs, which are being set as data. Has it been tried out? Is it being tried out? Yes, it is. And probably some of you listening now are coming from these countries here and hopefully have used extensively the machines already and you are happy with the machines. You can see we have research sites in Ethiopia and in Malawi, and you will see some data on that. We have the headquarters in South Korea, and we have regional offices in Switzerland. And machines are located in UK, Germany, France, Spain, Italy, in Europe. They are in many countries on the African continent. They are in Southeast Asia, we have machines, and we have in Central America, in Brazil, Panama, and Mexico. And as you will hear later, we have machines at the United States. So why shall we use a MELAB laboratory device? Why is that good? It has a benefit for the users and for the patients. It enhances the efficiency of the malaria diagnostics by streamlining the workflow. It reduces the manual tasks by enabling faster results, lowering the reliance on, as I said before, skilled personnel, which is not always available. And it supports remote view, which in my opinion is very important because you may not have the specialist close by, but you may have a specialist, via internet somewhere a few kilometers away. And you can send over the pictures and say, look, is that a malaria parasite? And a specialist can say, yes, it is, or it is not. So it can improve operational efficiency and also reduce the costs and promote a more sustainable, environmentally conscious diagnostic process. So sample to answer in 15 minutes, remote review capabilities, reduce the burden on skilled personnel, reduce the manual tasks, save the operational costs, and provide a sustainable process. So that's how we see it, how the machine could actually help you. Okay, so Unit 8 Report 2022 introduced the MELAB as an innovative product that overcomes the limitations of current malaria diagnosis. It's not only us having seen how limited these manual laboratory microscopic diagnosis is, it's also recognized by Unit 8. And therefore, they see the most advanced digital microscopes are fully integrated benchtop platforms such as Site Diagnostics or NOUL. So there are actually benchtop microscopy possibilities. NOULS is very much advanced with the automatic staining and also, very importantly, with the artificial intelligence-based analysis. NOUL has done a strategic cooperation with LabCorp in the United States of America. It's a Laboratory Corporation of America Holdings, and it's the largest diagnostic testing company in the US, and it's a global recognized leader. It plays a pivotal role in the diagnostic ecosystem as key validation hub for innovative technologies. And LabCorp has recently engaged in a collaborative project with NOUL and has already been published, a paper published in the Journal of Clinical Microbiology in 2025 in January. In one single note, the results of these studies, which has been done, they tested traditional microscopy and the MELAB device on laboratory basis in the United States. The sensitivity for microscopy was 81.8%, specificity 100%, positive predictive value was 100%, negative predictive value was 99.5%. With traditional microscopy. If you look at the figures for MELAB, everything is 100%. So they had 100% sensitivity and 100% specificity. This is more than I would have expected. It's a wonderful result, but of course it's on a small sample size and it is on laboratory in a laboratory environment. It's done on a laboratory environment, so and non-endemic areas. Okay, anyway, it achieved a high performance with 100% across all performance metrics. But how does it look in the field and why is MELAB? So this is now just quickly, I forgot to say that this is now the reference which we have seen with MELAB. So these are the images MELAB has seen. This was a very tricky one here because it was initially classified as negative by the manual method, but then when you look at the MELAB method, it clearly is a parasite, which you then can see on the result screen, but also on the viewer. It clearly is a parasite and would have been overlooked by a classical microscopist. But how does MELAB perform in the field? So that's probably the most important data. is actually good for it. And I would like to start here with a quote from Professor Christian Köpfli, who is an assistant professor at the University of Notre Dame and is a close collaborator with NOUL on field work. So he quotes, patients can be treated quickly with accurate diagnostic tests. There are many cases of finding malaria-infected patients on the MELAB that local experts have not found in Ethiopia's clinical settings. Because MELAB is easy and simple to use, it's a must-have product in the clinical field. But how has it performed? So there have been studies done in Ghana and in Ethiopia together with Christian Köpfli in 2022. It was a study with the University of Notre Dame, USA, KNUST in Ghana, and the Gondar University in Gondar. It was a research collaboration, and the test methods which compared the MELAB device, RDTs, microscopy, and PCR. And the results have been presented 2023 at the ASTMH conference and 2024 at the MIM conference in Rwanda, and recently have been published on the PLOS Global Public Health. So there were 659 febrile patients enrolled in Ethiopia and 991 patients in Ghana. MELAB reached a sensitivity of 94% and a specificity also of 94% for both B. falciparum in Ghana and in Ethiopia. The sensitivity for P-Vivax in Ethiopia was 1997. In Ghana, there is no P-Vivax. That's why we couldn't get the data. And MELAB diagnosed 51 or 52 P. falciparum infections, which had an HRP2 deletion. I mentioned that before, the HRP2 deletion becomes now a major threat in Africa because parasites lose the coding gene for the histidine-rich protein 2, which is actually the target of the rapid diagnostic tests. And if you lose the gene and you cannot express this protein, it is, of course, lost. And these patients would have been diagnosed as negative, falsely negative, because they have an infection, but the parasites don't have HRP2. So these are already very good data, and I think compared to microscopy, we're reaching the same way, but with a much more convenient, efficient, and much more reliable way. Another study done also with Christian Köpfli, it was also in Ghana and in Ethiopia, 24-25. Again, Notre Dame, KNUST, and Hawassa University and Gondar University, both in Ethiopia. And it was a sponsored research collaboration, again, comparing MELAB, RDT, microscopy, and qPCR. Here we had 2,201 samples in Ghana and in Ethiopia. And the number of samples was confirmed, the number of positive samples was confirmed by qPCR of 475 cases. So MELAB reached a sensitivity of 96.3% and a specificity of 19.8% for falciparum. Sensitivity and specificity for P-Vivax was 96.8 and 97.8. And MELAB correctly identified the species in 99.3% of falciparum and 96.5% of P-Vivax monoinfections. This also has already been or is about to be published. It can be found in the medRxiv 2025 AI-supported automatic microscopy for malaria diagnosis. So it will be published soon in one of the peer-reviewed papers. We have also done then a validation report, validation studies, and one was done in Nigeria. 400 samples were collected and analyzed with MELAB and 2 expert microscopists, and here the results were 94.4%. Sensitivity and specificity was 98.1, positive predictive value 96.6, and negative predicted value was 96.9, sorry. This was presented as poster in 2024 in Rwanda at the MIM conference and orally at 2024 at the ASTMH meeting in New Orleans. Then there was a pilot study in Spain with the Spanish Malaria Reference Laboratory, ICTMM. This is now a developed study in a developed country, in a rich country, of course. MELAB results and showed all the expected positive falciparum and vivax cases. There was a 92% agreement with thin blood smear microscopy, sensitivity 93.6 and specificity 89.1. But 8 out of 11 MELAP positives, but microscopy negative samples were true positive by PCR, which then corrected the sensitivity to 94.4 and the specificity to 97.2%. And again, this was presented at the 2024 ICT-MM conference. And finally, last but not least, a technical paper from the MELAB published in the Frontiers paper. This was by one of our Korean colleagues here, Bea. It showed a sensitivity and specificity for P. falciparum with MELAB, a sensitivity of 95.2% and a specificity of 91%. In Malawi, with a sample number 488, there was a high agreement between MELAB and traditional microscopy, sorry. And the Pearson's coefficient is 0.96, and the ROC curve indicates near perfect classifier accuracy with an area under the curve of 0.99. All right, okay, so this is what we can deliver you as information on the performance of the machine. Now, the big question is, what is our value proposition? What makes MELAB so valuable? And this is my opinion, but I share that with the company. I think it provides a high diagnostic accuracy. It achieves reliable results with a simplified infrastructure of just a compact device and cartridges. It provides, well, let's go here. It has an automatic workflow. It minimizes human error through the AI-driven analysis, ensuring consistent diagnostic quality, which I think is really one of the highlights of the machine. It gives you a rapid on-site result, about 15 minutes. It delivers immediate results, securing the golden hour and supporting fast decision-making. And finally, it's standards-compliant innovation. It meets the gold standard microscopy requirements while it enhances the efficiency through innovation. And I hope I could a little bit convince you that the machine is at least worthwhile looking at it, and perhaps some of you have used it, can confirm the whole thing. And thank you very much for listening. And now we are moving on to the Q&A session, and I think I hand over to Jack, who probably will then moderate the Q&A session. Thank you very much.
Thank you very much to you, Professor Beck. It was an excellent presentation and very comprehensive.
You're welcome.
So, as Professor Beck mentioned, I will now move into the Q&A session. So we have quite some questions, 6 so far in the Q&A box, and we got also one in the chat, which we can cover at the end, I think. So I will start with the first question. More than being known as a gold standard, microscopy is well adapted to intermediary and peripheral level of healthcare system in South Africa. Is the NOUL system, or could the NOUL system be deployed in that part of the health system? What are the minimum infrastructure needs for operation?
All right, Jack, do you want me to answer that also? I could do
If you
so.
like, yes, please.
I know a little bit about the South African infrastructure and the South African health system. In principle, I see no problem that it can be included into or integrated into the system. So, because it provides the same data as you would have with a classical microscopy, as you mentioned, which is well integrated in the health system in South Africa. And the data the machine provides you are exactly the same as a microscopist would provide you. The only thing is most probably a little bit more reliable and a little bit more faster. In terms of infrastructure, what do you need? You need a power socket. You need a laboratory. You need a handheld pipette to pipette 5 microliter of blood. And you need, of course, a blood lancet and all what you need for bloodletting. And if you have that, I think that's all. The only thing you need, this is alcohol, ethanol alcohol. This is something you need for the fixation on the machine. But this is available everywhere in the world, as I know, and is particularly available in South Africa. And even in the remote places, I think you can very well integrate this machine into the South African system. I do not see a problem. What I would like to mention is you need the cartridges. Besides the instrument, you need the cartridges. And there's always the discussion, what is the shelf life of the cartridges and what is the storage condition, which of course for Africa is a big problem. So shelf life has been so far 2 years. So you have a shelf life and it is at room temperature. Room temperature, hopefully not exceeding 35 degrees, and it should be a reasonable dry place. But because it's sealed, it's not much a big problem, and it's in an alum, so in a dark seal. So even if light shines on it, it doesn't matter really. So I think it could well be placed into a system where you just store your cartridges. I hope that could answer the question.
I think so. Thank you very much, Professor Beck. And the next question is, I think, coming from Nigeria. And the question is, how can the diagnostic solutions be made more affordable for those in low-income countries like Nigeria? Would you like to take that one, or shall I take it?
I think I try my best. I definitely have no solution, and as I say, I'm a scientific advisor, and I see the problem with costs of cartridges, instruments, and so on and so on. And I'm not quite sure how the company itself, NOUL itself, will strategically place itself here, whether it will be on an abonnement, for example, where you just take the cartridges and you get the machine for free, but you have to pay for the cartridges. Uh, you have somebody like USAID. I know this is a big problem currently, but you know there are models where you can get support for the machines, you can support for the consumables. I don't know, and I don't want to hang out of the window here, and I rather give it back to the company to make a statement how they want to make it more affordable for the customers in low-income countries.
Well, so maybe I can give a little bit of an answer to that. And of course, we will not discuss the exact pricing in this webinar. So I would like to emphasize that anybody who is interested in getting exact quotes to contact us directly. But in general, of course, as I mentioned at the very beginning, so our mission is really to make diagnostics more accessible for all. And of course, one part of this is actually to control the costs, especially since for malaria, we are mostly talking about endemic regions in low and middle income countries, so to speak. So from a design perspective, the device and the consumables have been designed to be as low cost as possible, but without sacrificing any of the diagnostic accuracy and precision required to fight these diseases. There are initiatives, of course, that we can take to reduce costs, and costs, of course, depend as well on test volumes. So in endemic regions, of course, we have much higher test volumes than in non-endemic regions, which of course can affect the pricing. Um, yes, but additional to that as well, I think just in general having a lower cost design in relation to other competitors is something that we should look into. And on top of that, we should not always talk about just the cost of the instrument and the consumables itself, but we should also look at the savings that it makes when compared to the manual method, which also have costs associated to it. Hope that together with Professor Beck's answers to the question there. Okay, the next question is from Katja Kupke, and the question is, are all malaria parasites detected and differentiated? Plasmodium falciparum, Plasmodium vivax, Plasmodium malaria, Plasmodium ovale, and Plasmodium knowlesi. I think this is one for you, Professor.
Okay. Well, it's not an easy one, but I can answer. The quick answer is no. So the machine has been trained for Plasmodium falciparum and has been trained for Plasmodium vivax. So these are being recognized as such. If you have other parasites, they will be recognized as a damaged cell or as a different cell. So you will get either it's being falsely called a Vivax or falciparum, which I doubt. But in principle, it will be shown as a damaged cell, and it will pop up on the screen, and it will ask you for review or action required. So there will be something where somebody or the machine tells you, I don't know, please have yourself a look what it is. And then you have to go over either by the viewer or by the little screen you have on the machine to identify it, but it will not tell you anything because we have, slightly honestly saying, we don't have enough samples to train the machine either on malaria, ovale, or nosai because there are too few samples available. And you need to really have a good training material. You need thousands of images which you can use. And unfortunately, they are not available, probably because the parasites are so low. And what I need to say here, of course, is, none of these ones, except nosai, I have to admit, except nosai, neither malaria or ovale is a deadly disease and, can be um, is not killing. So, but of course, needs to be identified. There's no question, but there's no rapid diagnosis needed. But nosite it's different. Nosite is extremely dangerous, as you probably all know. It kills rapidly, and maybe it will be recognized quickly on the machine as something you should have a action yourself. Does it answer the question? I hope so. The machine definitely is not trained for it.
That's correct, Professor Beck. Thank you very much for the answer. And just for those of you wondering, if you do find an ovalia, malaria, or an ELISA, the system will flag that there is a positive infection, but it will not know the species. And there is an
Mhm.
option for the user to manually validate the species themselves. And just while we're talking about species, because there is some related questions about species in the chat. One is from Dan Helling. There is another also again from Katja Kupke, and I believe there is also one in the chat. And essentially, the question boils down to what about the detection of mixed infections?
That's a tough one. That's a tough one, and unfortunately, to my knowledge, mixed infections may not be labeled. You will find all the parasites. As I said, every single parasite which is detected by the machine is being displayed on the side of the display or on the display itself or on the viewer. So every single parasite which is being found by the machine is being displayed. But the machine will either say P. falciparum, which most probably will be the most frequent one, or P. vivax, if that's the most frequent one, but it will not tell you a mixed infection. So what I would recommend is, as if I would be a user of the MELAB, I would definitely recommend to have a close look at the parasites which are displayed. And if I see, okay, well, this looks different, this could be a wale or something else, then again, as Jack just said, then you have the manual way of changing and you can actually put in your opinion. But it does not highlight and does not flag mixed infection. Maybe at a later stage, but I think in the moment it is not done. So you don't get a highlight of mixed infection, unfortunately.
Thank you very much, Professor Beck. So the next question is, what are the current challenges regarding AI automated microscopy?
Well, I'm a biologist and not an AI specialist. The only thing I can say, I think it's a number, it's probably the number of images a machine can be trained on. But perhaps I see you nodding, Jack, and I see you nodding as well. So perhaps you can give a better answer, please. So when perhaps
Yeah,
you,
sure.
I don't know.
Sure. So, I mean, the main challenge with the artificial intelligence is, of course, training data sets. So, when it comes to, for example, if we wanted to make available the AI speciating malaria, ovala, and Nolisi, we would need samples for that. And generally, we need a minimum of 200 samples. And the more, the better, of course. The more data you can give the AI, the better. And of course, some of these species are quite rare. Um, I'm based right now in Europe, and of course, it's very rare here. But even in some endemic settings, some of these species are quite rare. So actually, the challenge is to gather the data and train the AI. And I think another challenge that needs to be thought about when we do train the AI is that we also don't train it to make false positives as well, which can cause, of course, a lot of extra workload for the user. So we really need to be careful when we choose our AI thresholds in order to not ensure that, for example, if you have morphological inclusions of RBCs like Hal Jolly bodies, for example, or general stain artifacts, that they do not trigger a positive result for an infected RBC in a sample that is otherwise plasmodium free. So I think it's always one of the big questions in science, of course, is to balance the sensitivity and specificity. And this is something as well that applies to artificial intelligence. So I think those two areas are the big challenges when it comes to AI automated microscopy. Hope that answers the question.
So
Okay.
everybody who has a lot of noseye species could try to have a collaboration with NOUL, because that is definitely a
Absolutely.
website which is worthwhile looking at.
Absolutely. Absolutely. Okay, so we have Dan Pelling again, and he also said, lovely talk, thank you, just as a little feedback there. And his question attached to that is, are the sensitivity and specificity data applicable across all stages of parasite development?
Well, as you well know, Plasmodium falciparum usually has only the ring stages shown, so you only get the ring stages. And here, the sensitivity and specificity data are, of course, everywhere the same, more or less. Of course, there are fluctuations, but they are the same. Gametocytes, again, we have to say we haven't done any studies, to really say, or I don't know of a study that been done by NOUL for gametocytes, again, because gametocytes is quite comparatively rare event to see on the slides. So you probably don't have the training material to make a good data set on that one, and a good statistics, I cannot say. With Vivax, I think Vivax, it's applicable to all stages because it recognizes all the Vivax stages. And, regardless which Vivax stage is recognized, it's being counted under sensitivity and specificity as long as it recognizes it. So this is, to my knowledge, yes, it goes across all stages if all stages are available, except for gametocytes.
Thank you very much, Professor Beck. So the next question is from Sylvia DeSanti. And Sylvia asks, thank you for the presentation, first of all. And how were the tests conducted to determine the limit of detection? I guess maybe this is one for Null or Professor Beck.
I
You're
think
aware
that's
about
probably
that?
no, because I wasn't part of it. It perhaps, for NOUL to identify that or to say how that has been done. So I wasn't part of that.
Thank you. So I have to be quite honest here. I'm not myself 100% sure. My guess would be that we did serial dilutions of a malaria sample to reduce number of the number of parasites in the sample until we get to as low as we can go, so to speak, the limit of
Mhm,
detection,
mhm,
as it
mhm.
were. But I don't want to give that as a 100% answer because I'm not 100% sure myself. So, Sylvia, we will come back to you actually with an answer for that one. And thank you very much for your question. It's indeed a very good one. So the next question is again related to sensitivity and specificity. This is from Jyoti Panasar. And Jyoti asks, with regard to the sensitivity and specificity, was this calculated after any reclassification of cells by a user?
Once again, I think I must pass out here because I wasn't part of the study. So I don't know. I would assume yes, but I don't know. I honestly don't know. So perhaps, again, you have to ask, those who have run the study and should probably come back to you later. So that's that I would say. I would say it is done like that.
Thank you, Professor Beck. I believe most of the studies were before reclassification of the cells by a user. Some of them may have been done after a reclassification by a user. So, because of course the you're asking then a different question. So you're asking the sensitivity and specificity of the device. And if you are taking the reclassification of the user, then you are talking about the sensitivity and specificity of the device plus the user, actually. So if we talk about real device performance, exclusive of which user is using the device, then we should do that before reclassification of the cells by the user. But yes, we will come back to you with a comprehensive answer on that, Josie, because honestly, I'm not sure on all of the studies which ones were
Mm.
reclassification and which ones were pre-classification, so to speak. Um, Then we have another question from Dan Pelling. Have there been any studies comparing output in Babesia cases? And Professor Beck, would you could you answer this one?
The, well, I think the straight answer would be no. No, there has no. To my knowledge, there was no comparison with Babesia cases. But you open here a whole can of worms. You know that could well be done for the veterinary part or even for the human part. But I don't think everybody has looked into that at all. At all. But you're absolutely
Yeah,
right. It would be fantastic for the veterinary diagnosis as well.
yeah, fully agree. Fully agree. Okay, so the next question is from Gabriel Montoya. I hope I pronounced that correctly. Considering the temperature, climate, and humidity typical of tropical countries, specifically in the context of the Amazon region, which is an endemic area for malaria in Brazil, has the equipment been validated under these conditions? Is there any interference with the reagents or with the slide staining?
I think it has been done. It has been done in the humid chamber. It has been done in the warmth chamber. And as I said during the talk also, I think the shelf life is 2 years. And this is under ambient temperature up to 35 degrees. And humidity is not an issue because they are sealed. So under 5 minutes, when you take, or about the 5 minutes when you take it out, put your drop of blood in and run it in the machine. I don't think that humidity plays a bigger role or so. But I know, for example, in Ethiopia, we had also rainy days and it was sort of humid. Probably not like in the Amazon, which I know very well, but it didn't affect it, honestly. And I don't think it will affect it because everything is very well sealed and the machine is there's no problem, I think.
Yeah, fully agree. Thank you, Professor Beck. And the next question is from Mike Lyle. So Mike asks, there seems to be quite a lot of variability in specificity and sensitivity, sorry, between the various studies performed in different countries. What is the reason for this? For example, specificity of 91.4% in Malawi versus 98.1% in Nigeria.
This is a very difficult question to answer. First of all, the Malawi samples have been collected much earlier, so it may well be that the AI version which has been used was a much earlier version. Nigeria has been recently done, so it may be because there's also an improvement in the AI version. As you can see, it was also stated on the slides which version, at least in one case, it was stated. That may be the case. Um, Otherwise, no, I have no further explanation. I don't know. So, because the machine has not changed much, except the AI version, and the microscopists change, but that should not affect, the specificity and the sensitivity. So, I have no idea. I cannot tell you. It may be because it was earlier, because there's development going on. That would be my explanation. But perhaps you have a better, Jack or Gwen.
No, I think your answer pretty much covers it. And I think it also depends on the comparative method that you are comparing to is the gold standard. And then, of course, you will get different sensitivity, specificity, and
mm,
also interoperator variability between microscopies, as you mentioned.
mm, mm.
These are all factors which contribute. And also, of course, the level of parasitemia can also contribute, I
Yes,
suppose.
definitely. Yeah.
Um, okay, so there's a few more questions coming in. How is the analyzer controlled? Can you explain more about the staining? That the stain does not look like a typical Giemsa stain. Could answer this if you like, Professor Beck.
No, I can't really answer that. It's a modified Giemsa stain. It's based on fairly old stain, but it gives similar quality of stains. But perhaps somebody from NOUL who was involved in that could answer that.
Yeah. So the staining is a modified Romanowski-based stain,
Okay.
consisting of methylene blue, eosin Y, and a buffer. So it's the same components of your classic Giemsa stain, actually. Of course, depending on what staining you are used to, what staining protocol, also different manufacturers of stain, you might be used to slightly different results. It can also depend a lot on the pH of the buffer that uses. Of course, if you go to the more basic end, everything becomes more bluish. If you go to the more acidic end of buffer, then everything becomes more reddish. In my general experience in the field of malaria smears and peripheral blood smears in general, there is a huge variability in the preferences of the user with regard to staining. And of course, we had to standardize that as part of having a cartridge-based system, which means there is no variability in the stain from one cartridge to the next, which of course means there's a knock-on effect that some users may find that it is slightly different to the staining that they are used to. But as with all stains, is of course a slight change in coloration. Can be subjective as long as, of course, you are highlighting those important morphological features, and can just be a matter of getting used to a new pattern. Essentially, I hope that answers your question, Zara. Um, okay, so we've got a couple of questions about whether the AI continues to learn through use. I can take over that one and just answer it very straight. The answer is no. Because of course, otherwise we will not have a standardized algorithm. So all the AI is trained in-house, and then the algorithm is loaded onto the device pre-trained, and the algorithm on every device should analyze every slide in the same way. Otherwise, we lose standardization. If you imagine we installed 100 devices across the globe, and each of those 100 devices is learning depending on the samples and staining and sample preparation that is put through the device in each of those 100 sites. And then you took those 100 instruments back after 1 or 2 years and did some tests, you will get very different results between the device. So this is no way for us to ensure standardization. So no, the AI is not learning through use. However, I can say that if you have some interesting samples, some other Plasmodium species that might help us to train our AI, then please get in touch because that's definitely something that we are interested in. Um, okay, so let me just check that I've covered everything there in the chat. Detection of mixed infections, we covered this all as well. And detection of other parasites like trypanosoma. I guess this also goes into the direction of the earlier Babesia question. So the device is not possible to detect Babesia, trypanosoma, other blood parasites. Of course, the technology has the potential to do so, but that requires, of course, some R&D developments and investment and of course also data to train the AI. So this is something that, of course, we might look into in the future. Then there's a lot of questions that are all very similar, which is about the affordability of the system. So actually, I would just like to wrap up these questions very quickly. We do have various ways in which we sell the devices. So, for example, reagent rental, capital sales. So and so forth. And this is all things that we would discuss with you individually. So if this is something that you would like to explore further, please get in touch with us. Um, and with that, I would actually like to close the Q&A session as we are running a little bit over time here. First of all, I would like to say thank you very much to Professor Hans-Peter Beck for your time and excellent presentation. It was an absolute pleasure to have you here, and we really look forward to continue working with you in the future as our scientific advocate.
Welcome. Well, goodbye, everybody, and thanks for listening.
Thank you. And to everybody else who joined us today, wherever you are in the world, I would like to wish you a nice rest of the day, a nice evening, or a nice sleep for those colleagues over in Asia. Thank you very much for joining this first webinar of NOUL. Just as a short reminder, there will be a survey at the end of the webinar. And if you fill this out, we will provide you with the presentation and the recording afterwards. So please do that. It's really important for us to get your feedback to improve our webinars in the future. And with that, further ado, I will close the webinar. Thank you very much, everyone, again. Take care. Enjoy.
Thank you so much, and good night or whatever.